Good morning. It's now my pleasure to introduce our next participants, Dr. Eric Kelsick, CEO of Dyno Therapeutics, and Yvette Lung, Head of Corporate Development. So this fireside chat will be around 25 minutes, and the audience may ask questions during the session by typing them into the chat box in the Zoom window, and we can try to ask them on your behalf. So to start off, Eric, maybe you can give us an overview of Dyno's business model and its approach to AAV gene therapies.
Yeah, happy to. Thanks, Gula. I'm excited to be here today to share with you about Dyno. So maybe I'll start with our approach to AAV gene therapy. We're a company that's focused on AAV capsid engineering, and we'll talk in a moment about how that relates to the manufacturing challenge. But as you all know, in vivo gene delivery is the key challenge for gene therapy. And the vectors that we have from natural viruses are the best approach today, but they bring with them certain challenges, including manufacturing, but also other aspects that are the natural capsids aren't optimized for therapeutic delivery. So for many reasons, it's been difficult to engineer capsids to make them better. For example, in the past, we've been limited by focusing on only the quality of the different variants that we test or the quantity via, for example, rational design or directed evolution. And what Dyno has created is an AI-powered solution to bring all sorts of new technologies to the capsid engineering problem. I'll walk you through those different steps, things that we've added to the more traditional protein engineering workflow. So the first thing which is new is that now we have the ability to synthesize a very large number of sequences in high throughput, hundreds of thousands at a time, which enables us to build synthetic capsid libraries. We can also then apply next-gen sequencing to read out capsid property measurements in the most relevant animal models. For example, Dyno, we're focused on testing capsid properties in non-human primates because that's more relevant for translation to humans. And that generates a huge amount of data. For example, in one animal, we can test hundreds of thousands of variants in all the major organs. So what do we do with that data? We also automate the analysis of that by using machine learning. And that's very powerful because with a machine learning model, we can learn a lot about what types of sequences make capsids better. And then we can query that model to ask about new sequences that have never been tested. And we can query it many times in silico, choose the best variants, and then put those back into another high throughput screen, again, programmed by DNA synthesis. So that's a very new approach. It's in a technique that we call machine-guided design. Dyno is one of the leaders in this field. And this is an approach that myself and a team of researchers and George Church's lab at Harvard developed going back to 2015. Since 2018, we've been applying this now to capsid engineering as a company, as Dyno Therapeutics. Some recent highlights of our growth, for example, last year, we closed our Series A financing led by A16Z. That was 100 million. That enables us to really scale up the company. We're currently around 80 full-time employees and still growing. And we're fully focused on solving this problem for gene delivery. For example, working across liver, muscle, CNS, and eye. To bring it back to our business model, Dyno is a company that was built to partner. So we offer licenses to our gene therapy platform, to gene therapy product developers. Importantly, we're not developing our own products. Instead, we're working together to multiply the effectiveness of the whole ecosystem of gene therapy developers, because that's the way that we think we can help the most patients, maximize their impact, and also because it enables us to specialize on these cutting edge technologies and offering the best benefits of that to the entire field. So we would love to partner with all of you and would love to ask other questions in terms of how we might be able to work with the entire ecosystem of gene therapy investors.
Great. So just on the topic of partnering and the business model there, can you expand a little on to what extent discussions of manufacturing might play into your interactions with partners and how that fits into those dynamics?
Sure. Manufacturing is definitely one of the key elements that our partners are interested in. They're also interested in other properties such as tissue tropism, immunogenicity, as well as packaging size. And these really comprise the kind of capsid wish list, or what we call the ideal kind of capsid profile for these partners. So for instance, we can build in manufacturability as one dimension of that capsid profile, to really ensure that the capsids that we're providing to partners are as good or better than the starting serotype in terms of production yield.
Got it. And in terms of the structure of the partnerships, for the ones that you have, are your partners typically responsible for the manufacturing once you hand over the capsid, or what is the view on that currently, and do you see that evolving in the longer term?
Yeah, so Eric alluded to this a little bit earlier, but we're really focused, in terms of all of our efforts on solving that gene therapy delivery challenge. So we provide those novel capsids to our partners, and then they then combine those capsids with payloads to then clinically develop products inclusive of manufacturing. So we've been really lucky thus far to work with partners that have an incredible amount of experience in product development as well as in manufacturing to really bring these new therapies successfully to market. And like Eric mentioned, we're really excited to partner with additional folks as well.
Great. And Eric, you mentioned this earlier, but maybe we can expand on this a little bit more. You talked about how Dyno's AAV design capabilities can be applied towards increasing manufacturability. Can you expand a little bit on what that means and how this might set you apart from others in the space?
Yeah. Well, manufacturability is just one of the properties that's important for optimizing AAVs for gene therapy. But it's obviously a critical step no matter where the AAV is going, which tissue you're optimizing it for, how selective you're trying to make it. Everyone needs to manufacture the therapy. So that's one thing that we select for as part of our screens. What's really exciting about the new technologies is that we're generating so much data. It enables us to do the selections across multiple properties because we just develop an assay for each of the different properties. And then computationally, we can filter all the sequences to choose the ones which are going to be optimal for a given capsid profile. What's really interesting about AAV proteins, they're one of the most complex proteins to engineer, and that's because, for example, the capsid is formed from 60 different monomers that assemble together. So there's just a lot about the capsid protein, which is more complex than, say, a single protein that you might want to engineer. And that may be the reason, or maybe it's some other reason, but in any case, it's an empirical fact that most changes that you make to the capsid sequence will break the assembly and the packaging of the genome. So if those things are broken, then it's very difficult to manufacture the capsid. So that's one of the things that we screen for as part of our screening. It's the first step, looking which changes we can make, which might offer the potential for enhanced function, say going into a new organ, a new cell type. But then we screen those for the ability to assemble into the capsid and to package the genome. So it's one of the properties that we always measure. And a challenge with AAV engineering, because as you see, most protein changes will break the function, including manufacturability, is that a lot of engineered capsids have been more difficult to manufacture. And the reason makes sense that if you're selecting for all these different properties, for example, you select for going into a new organ, most likely it's also going to have a deficit when it comes to manufacturability. So this ability to select for multiple properties is one of the things we can do to ensure better manufacturability of engineered vectors. There's other ways, though, that it can help too. And that comes from enhancing the other properties. For example, increasing the efficiency of delivery into a target tissue. That can also help for manufacturing because, say, we make it 10 times more efficient. Well, now we can treat more diseases because we can reach more of the cell types that are going to benefit from the therapy. But we can further increase the efficiency still, say from 10x to 100x. Now with a 10x lower dose, we can just as effectively treat the patient. So that's better because the products are better. For example, it's going to be more effective, it's going to be safer, and it's also better because when it comes to manufacturing it, now you can make many more doses with a single batch, or the cost of goods for a single dose is much lower. So both therapies can be made more affordable for the entire payer ecosystem, and also profit margins can be higher for gene therapy developers and manufacturers.
Got it. That makes a lot of sense. And so with regard to some of the topics you just mentioned, the safety, the doses per batch, et cetera, in general, AAV gene therapy safety has remained a topic of discussion, especially after last year's FDA panel that was convened on the topic. Can you expand on additional elements that your capabilities could bring to the table in terms of improving safety and manufacturability along those dimensions?
Yeah, I'll just basically keep going down the list of things that we can improve about AV vectors. And, the reason why there's so much potential for improvement is that natural vectors weren't optimized for all these purposes. So we've already talked about ensuring that you can manufacture the vectors when you make a change to an engineered variant, about how we can make them more efficient, which lowers the dose that's needed. And that's one direct way in which we can make them safer, because most of the challenges that we've observed or the safety issues with AV to date have come from high dose AV. And the reason why companies are using high doses is just because the efficiency is so low that that's the only way that they believe there's a chance for the patients to benefit from the therapy. So people have been kind of doing as much as they can in order to give patients a chance at receiving some benefit, but that comes with risks. By making the delivery more efficient, you don't have to take those same risks. A much lower dose would be sufficient, and that has a big impact on improving the safety profile of a gene therapy. And then there's a third property too, which is the specificity of delivery. So efficiency is going into the target organ, target cell type. Specificity means that if a vector is highly specific, it only goes to that target cell. And for example, avoiding off-target cells, either in the same organ or even in other organs. For example, we could go specifically into the muscles, skeletal muscles, say, but then avoid off-target organs like the liver. And that could really dramatically improve the safety profile, both the indirect change from avoiding the liver and also the direct improvement from being able to treat patients at a much lower dose.
Okay, that makes sense. So some of the other factors relating to manufacturing that have been discussed with regard to safety have been things like full empty ratio and just the extent to which the capsid assembles the genome inside it correctly. And so you touched on this a little bit with regard to looking at assembly of the capsids that are produced by your platforms. But are there other elements that you can look at to address some of these issues, or other elements of manufacturability that you can look at that you haven't already listed?
Yeah, we'd love to learn more about what are the most important aspects of the manufacturing process that new partners would like us to optimize for. It's quite a powerful approach because, as I said, we can integrate any new assay, any dimension into a target capsid profile. So to date, we focused first on just the manufacturability, which is the assembly and the packaging of the genome. For example, with a full empty ratio, it happens that our assay, because it's based upon measuring of DNA sequencing, it directly selects for capsids which are packaging the full genome, for example. However, there's other aspects that we could add into a profile. For example, we could optimize for aspects of purification of the capsids, or the ways in which they are extracted or lysed from cells. So those are all things that we're very interested in exploring and could potentially help a large number of players, both direct gene therapy developers as well as those who are focused on the manufacturing parts, for example, CDMOs.
Great. And then I know you mentioned you're not internally focused on doing the manufacturing yourself, but one of the other things that people look at with regard to manufacturing, as they think particularly towards a commercial scale or commercial phase, is the scalability of the process. So to what extent is that something that you think about in terms of the manufacturability that you screen? Because I imagine different types of processes, when evaluated, can have different scalabilities.
Yeah,
go ahead, Yvette.
Oh, I was just going to say, I think it's definitely an important part of kind of those core properties of that profile that we've been talking about. And one of the really cool ways in which we've been able to apply our machine-guided design here is that we do a lot of in silico screening. So we'll go through millions of variants before we get to the hundreds of thousands of variants that we then move into in vivo experiments. And within the in silico screening phase, we really select for capsids that produce well in order to move into the kind of in vivo phase. And I think that kind of ability to continue to move through that high throughput screening allows us to really select for better and better capsids ultimately that kind of pop out of the engine.
Eric, were you going to say something also?
No, Yvette said it all. Thank you.
Great. And so we touched a little bit on some of the challenges with regard to Aed manufacturing that you're thinking about, but maybe we can elaborate on that a little bit more. Are there particular challenges within the Aed space that you've noticed that you're hoping to address, or that you see as representing current bottlenecks within the space?
I can start with kind of a big challenge. I think there's a huge issue with scalability, obviously, but across multiple different ends. So first, we can't really kind of potentially reliably reproduce results across systems. So as we think about moving from adherent into suspension, and then as you think about kind of the smallest to the highest ends of batch sizes, we've really been operating largely within the kind of 100 to 10,000 liter scale bioreactor productions today. And then last but not least, as we think about capacity as well as supply constraints, we've all been kind of suffering from the same plasmid and material shortages. So really being able to scalably do production in light of all of those has been certainly challenging for all of us. And one of the ways that we've been thinking about it is just, building out operational excellence. And we're lucky to be able to focus on that research grade AAV production. But as we think about CDMOs, as I'm sure many will talk about today, are building out additional capacity to service the industry. We're happy and excited to hopefully partner down the line to really meet that growing demand as we expand.
Got it. And so within that kind of ecosystem of biotech product developers, CDMOs, and you guys, can you expand on how you fit into that ecosystem and how you envision your role evolving? Your view.
I can start, and then we'll see if Eric has more to add. But I think of it as, this manufacturing and the scalability challenges are really kind of something that we need to kind of all tackle in different ways. And there's a lot of different approaches that ultimately will help us really achieve kind of the goal of scaling up better. So one of the avenues that we're pursuing, obviously, is to design novel capsids that allow for improved manufacturability, all of what Eric's mentioned already. But others in the industry are kind of focused on whether it's alternative methods of plasmid manufacturing or trying to optimize downstream purification methods. There's all these different approaches that are going to come together ultimately for us to help solve this. And so we're tackling it in a very specific way, and we're leading in kind of the machine-guided design approach there. But again, others are approaching it in different ways.
Yeah, and to add to what Yvette said, what's so exciting about this and talking as part of this group is there's multiple ways we can approach solving this problem, and everything we're doing on the capsid side is additive with other improvements that people might, do to the process development or manufacturing side. So real excited to be able to partner with others across the whole ecosystem so that together we can help more patients.
And are there additional important innovations around Aed manufacturing that might better enable your platform to meet or exceed its goals? I guess you touched on some of them, including optimized plasmid approaches, purification, things like that. Are there other areas that you see potential synergies within other approaches?
Well, I think there's still a lot about AV biology that we don't understand, and for that reason too, still huge potential for new innovations. So I especially would be interested to see what's coming out from the basic research for example, when we did some of the early work at Harvard developing our approach, we did a systematic scan across the entire capsid protein. And one thing that came out of that, which was very surprising, was we discovered there's this other gene there, which we called MAP, membrane-associated accessory protein, just a completely new gene that we didn't know about before, embedded right within the capsid. So it just shows you that we've been studying this system now 50 plus years, and there's still a lot of things that we don't know. More recently, we've learned that that's involved in cellular secretion, that MAP protein. So that might be interesting from a manufacturing perspective to somehow modify as well, just as we're modifying the capsid. There's still a lot of things, though, that we just don't understand about how AAV works. And I think once we do, that could be really guiding for how we could continue to improve these technologies. For example, how the capsid assembles, how some cell lines are better than others, and why is that? What are the factors that are associated with production? And if we understand those, can we also engineer them to make them better? So this is also why Yvette mentioned, we see ourselves today as a leader in capsid engineering, but we're really investing in these fundamental technologies for sequence design. And all of gene therapy fundamentally comes down to design of sequences. So everything we're doing with the high throughput screening, the machine learning, and the automation of those processes can similarly be applied to other sequence elements, which is a direction that we intend to explore in the future.
That's really interesting. So to what extent can some of these machine learning and other approaches address some of the things that you just mentioned, like optimal cell line engineering as a producer cell line and things like that? Is that something that you think you guys would be potentially approaching, or is that another area where you see synergy with let's say CDMO partners?
I think there's always potential for synergy. So we really think about Dyno as an open platform where we are offering new innovations to our partners. But if they want to add to that innovations they're developing or also in licensing, we see that being very compatible with our goals to help patients. Fundamentally, though, there's few limitations on what we could engineer at the sequence level. All these things like the cell line or, for example, how the payload is expressed, other aspects that control, how it's produced, those are all sequences. So long as we can measure the differences, we can turn those into an assay and apply a machine-guided approach towards optimizing for therapeutic efficacy.
Mm-hmm. Got it. That makes sense. And so in the last couple of minutes, I kind of wanted to come back to a higher level question regarding whether there's any additional lessons that you've learned from the various CMC challenges that have been encountered by others within the space that you can apply towards your work with your partners.
I think we touched on a lot of it. I think really the need, as we've seen, for better kind of manufacturable AAV capsids really just hasn't been greater. And I think we're at an inflection point where we're seeing a lot of companies really focus on all of these different types of approaches that we've talked about, whether it's capsid engineering to thinking about the manufacturing side of things, focusing on different cell lines or different types of promoters or different types of, plasmid manufacturing options as well. And I think we all just have to work together ultimately to really get to the end goal. And we're really excited about the approach that we're taking. We think it's really unique in the space of what we've talked about today. And, like Eric just mentioned, our approach really can be applied across a lot of different areas and specifically to manufacturability as well.
Perfect. So I think we're just about at time. So I would like to thank you guys again for your participation and for the great discussion.
Thank you so much for the invitation and for the opportunity to speak and share more about Dyno.
Thanks for having us.